Hi everyone, this is George Steward, and today I'll be presenting my microcosm project for EVSP416 General Ecology. Beyond being an assignment, I was interested in building this microcosm to get a hands-on look at how a small closed ecosystem functions, especially using the materials from my own Central Texas environment here from my yard. I wanted to see the decomposition and nutrient cycling play out and observe which of the local plant and critters could adapt to the sealed jar conditions most efficiently. Ecologically, microcosms like this are valuable tools, and they let us test ecological theories, study species interactions in a controlled way, and sometimes even inform bigger environmental questions as discussed by researchers like Ives and others. Ives et al. 1996, Benton et al. 2007. Excuse me. My specific project involves a sealed terrestrial microcosm designed to mimic local conditions. Okay, my main hypothesis going into this was that the sealed terrestrial microcosm, once assembled with the local soil in my yard that you can see in the picture with my daughter, various plant cuttings, leaf litter, and small invertebrates, would develop into a somewhat self-sustaining system. I expected to see the signs of ecological processes like the decomposition and nutrient cycling, and I predicted that the hardier, drought-tolerant plants like the elephant bush and Smilax or greenbrier, along with decomposers like the isopods, would persist while some of the less adapted initial cuttings might die off. So my daughter and her younger brother helped me gather the materials for this microcosm. I built the microcosm using a large glass mason jar. I started with a base layer of soil collected locally here in Central Texas. Again, on top of that, I added the leaf litter, small twigs from ash juniper and live oak, cuttings of several other plants, including Portulacaria afra, which is the elephant bush, Smilax or greenbrier, and lantana, which is a native plant here. A few small rocks and a bird feather for extra organic matter. I also introduced some isopods or roly-polies. There was a millipede and likely some fly larvae present in the soil, visible from one side of the jar. Bluebonnet seeds were added as well, although they did not sprout. I added a good amount of water initially and then sealed the jar. I monitored it weekly, took photos, and I made notes and sketches of the changes, which I compiled in my log. Okay, let's look at how the microcosm changed over the roughly 7 weeks from early March to late April. Excuse me. Initially, it was a fresh mix of green and brown materials with high moisture evident from the condensation visible on the jar. Note the pictures will be viewable on the next pages. Okay. That was the week 1 image, and as you can see, the juniper is showing green, the Smilax here, which had no roots visible when I stuck the cuttings in the ground, nor did the elephant bush have any roots, but they're known to root fairly easily and maintain their green for quite some time. Here are the live oak cuttings with the lichen. These are goldeneye lichen attached, and it's still very green. There's the lantana in the center, just a cutting, no root, again, an acorn, and various dead material scattered around. So by the second or third week, some of the initial cuttings, like the juniper and lantana, started showing stress and began to decay, while the elephant bush and smilax remained healthy. This one at least. There was no new growth on this smilax cutting. Early signs of mold appeared on the decaying matter and the feather. Looking back, I realized that using only cuttings meant none of the plants had established root systems. They likely used significant energy trying to root, potentially disrupting the balance needed for immediate air and water cycling compared to using established plants. Around week 4, the system looked noticeably drier, although the hardy plants were still doing okay and the smilax was showing some growth. Fungal activity was prominent, and you can see, I think, some of that, and there was especially a lot on the feather, including a distinct fungal fruiting body that appeared briefly this week. Actually, correction, that was on week 5, as you can see here. There are the 2 sprouts, unknown origin. I do not believe that these are the greenbrier, the bluebonnets, some other type of seed. And you can see the millipede is still alive there in week 5. And the isopod, I believe, is visible. There's one. There are a few of those in here. Around the beginning of April, this is week 5, observed more dryness. I added some water along with the biostimulant that I use. The system seemed to respond well. The surviving plants looked healthy. Smilax showed more vigorous growth here. And decomposition continued actively. The fungal fruiting body that I mentioned there, by middle April, in week 6, the elephant bush and smilax continued to thrive, with smilax growth quite noticeable and some new shoots popping from the top of it. The mystery sprouts that had emerged earlier had died off, stuck to the glass. Decomposition continued, integrating the dead matter into the soil layer. And continuing on into week 7, the final week of observation, the system showed further shifts while the main plants remained stable. Millipede was no longer observed and presumed dead, though the isopods remained active. This suggests ongoing dynamic changes even as the system settled. More fungal mycelial growth there. Okay, so what are the main findings? The microcosm clearly demonstrated ecological sorting. The Central Texas natives adapted to drier conditions. The elephant bush and smilax and the isopods thrived while other plants didn't make it. And the millipede eventually died. We saw active decomposition, breaking down the organic matter. The importance of water balance was also clear. A key takeaway for me was the challenge posed by using unrooted cuttings. Their initial energy expenditure on rooting likely hindered the immediate establishment of effective gas exchange and transpiration cycles compared to using plants with existing root systems. And did this support my hypothesis? Partially. So, a functioning system with persistent life and signs of nutrient cycling did establish. However, the significant die-offs, the need to add water, and the potential initial cycle disruption suggest it wasn't fully self-sustaining. So the failure of the bluebonnets to germinate also points to the challenges of establishing specific species in these closed systems. And bluebonnets are nitrogen fixers. They fixate atmospheric nitrogen into the soil. It could be that there was plentiful nitrogen in the soil already, and they did not germinate for that reason. This experiment definitely leads to more questions. I'd be interested to test how different initial moisture levels might affect the outcome, or compare jars with and without the biostimulant that I added, seeing if the surviving community remained stable over a much longer period. That would also be valuable if I were to repeat this, setting up replicate jars to test specific variables, having a control, monitoring moisture more carefully, perhaps starting with fewer competing plants, or using a larger container could yield more robust results. Crucially, I would include at least one small established plant, possibly a type of small tree, with roots from the beginning to facilitate the immediate start of gas exchange and transpiration cycles. One that comes to mind is a desert willow. They're pretty easy to root from cuttings, and they do well in this environment and many environments. Very drought tolerant. Looking beyond this specific jar, microcosm studies are relevant to general ecology because they let us see fundamental concepts in action: limiting factors, adaptation, decomposition, nutrient cycling, and basic community assembly. As researchers like Ives et al. 1996 pointed out, they allow us to study interactions in a manageable way. And Benton et al. 2007 argued that these small-scale experiments are valuable for understanding ecological mechanisms and developing testing theories that can inform larger ecological problems. Acting as a bridge between the lab and the field, they provide a window into processes that might be harder to isolate or observe directly in complex natural ecosystems. So here are the references that I consulted regarding the role and relevance of microcosm studies in ecology. Thank you for your time, and I'm happy to answer any questions that you might have.